So let's continue our discussion of bacteria and archaea. Let's move on to some of the external structures that you'll see. We'll have some extracellular appendages. These are appendages that are on the outside of the cell, hence the term external. Some are used for attachment, some are used for movement. Flagella and axial filaments are always going to be used for movement. Sometimes an axial filament is referred to as an internal flagellum. Depending on who you talk to, they'll call it one, call it the other. To me, it's a potato-potato situation. For attachment, fimbriae and pili are going to be used, in addition to the glycocalyx. But the glycocalyx really isn't much of an appendage. It's more of an oozy capsule, so to speak. So, for our appendages, flagella are movement, fimbriae and pili are attachment. Let's talk about the flagellum. The flagella is, as I've mentioned before, multiple times already, we use it for movement or motility. And there's going to be three distinct parts or sections of our flagellum. We have the filament, hook, and basal body. So if we look, our basal body is going to be the part of the flagella that's embedded within the cell membrane and the cell wall. This contains the guts, the flagellar motor, so to speak. And then we're going to have a big bend in the hook, or a big bend at the end of the basal body. This 90-degree turn is known as the flagellar hook, and then finally we have the filament sticking out from the end. So if we look at a bacterial or an archaeal flagella, so a prokaryotic flagella, it spins in circles like a propeller. Here we can see a transmission electron micrograph of the basal body of a flagella. This is an awesome micrograph, I might add, by the way. A key difference between our prokaryotic flagella, so our bacterial and archaeal flagella, and the eukaryotic flagella is that a eukaryotic flagella will move that eukaryotic cell with a whip-like motion, whereas with a bacterial and archaeal cell, the flagella moves with a spinning motion instead. Depending on where the flagella is arranged or attached and how many flagella there are, we have some different terminology for their arrangement. When we say polar arrangement, I mean that all of the flagella are located on an end of the cell. If we have one flagella on one end of the cell, it's going to be a monotrichous arrangement. If we have a bunch of flagella all stuck on the same end of the cell, it's a lophotrichous arrangement. And then if we have flagella on opposite poles or opposite ends of the cell, we have an amphitrichous arrangement. Now, I'm talking about ends of the cell right now. So, would it be possible for these terms to apply to a cocci cell structure or cell shape, so that spherical cell shape, cocci? And the answer is no, because if we have a coxal cell, we don't necessarily have those nice, well-defined poles. But if we look at bacillar cells or rod-shaped cells, we have nice, well-defined poles that make it very easy to categorize our polar arrangements. We also have peritrichous arrangement. Peri is a prefix meaning perimeter, and with our peritrichous arrangement of flagella, the flagella are going to be all over the surface of the cell, randomly. So here we have the monotrichous. Here we have our lophotrichous. There we go, the term for lophotrichous. Here we have amphitrichous, opposite ends, and then here we have peritrichous. So, flagella exist to move the cell around. This process of moving the cell around, generally speaking, is going to be referred to as chemotaxis. All bacterial cells, and actually all cells that are moving around in general, are going to move in response to a chemical signal. Generally speaking, if it's a positive chemotaxis, it's going to move towards the chemical stimulus. If it's a negative chemotaxis, the cell is preferentially going to move away from the chemical stimulus. This chemotaxis process of moving towards or away from a chemical stimulus is going to be kind of rough looking. It's not a very pretty sight. It's not like the bacterium cell beelines it in a straight line towards or away from the chemical stimulus. Instead, it has kind of a random appearance to its movement. It's called a run and tumble movement. The run is when the flagellum is moving. So if our flagellum is spinning counterclockwise, the bacterium will have a nice, smooth, linear motion, and then, all of a sudden, the bacterium changes directions, and that's the tumble. And that's when the bacterium flagellum reverses quickly to stop the cell and change the course. Now, if the cell is moving in the direction of a favorable chemical stimulus or positive chemotaxis, those runs preferentially last a little bit longer. If the cell is moving in the wrong direction, it's preferentially going to have an increased number of tumbles. So it looks like this. We have the cell moving around in the straight line. It's a run, and then all of a sudden the flagella switches directions, and the cell tumbles around and reorients itself. If we have a peritrichous bacterium, the flagella will group together and preferentially spin in the same direction. And then for the tumble, all the flagella start spinning all over the place, and then the process repeats itself. If we look at this chemotaxis process, preferentially, the runs are longer in the direction of the favorable chemical stimulus and shorter when the bacterium is moving away from that favorable chemical stimulus. And the tumbles are whenever the bacterium changes direction. Now, if there is no chemical stimulus for the bacterium, then the runs and tumbles are just going to be completely random, complete gibberish. Those periplasmic flagella are kind of tricky to understand. They weren't very well understood for a very long time because, well, they were stuck in between the cell wall and the cell membrane. It's hard to find them. Any corkscrew-shaped bacterium or twisty-shaped bacterium is going to have the periplasmic flagella, so the spiroret-shaped bacterium. And these bacterium have a very wiggly mode of locomotion. The flagella is spinning and causing the whole bacterium to undulate its body for movement. Another name for a periplasmic flagella is an axial filament. These are going to be, as I mentioned before, an internal flagellum between the cell wall and the the cell membrane or cytoplasmic membrane. Some additional attachments that we have are the fimbriae and pili. A fimbriae is a very small structure that looks very similar to a cilia. These allow for adhesion between the bacterial cell and other host cells. Fimbriae are essential for adhesion and infecting host tissues. A pili is used for conjugation between bacterial cells. So this pili, or pilus, depending on if you have a plural or singular, is going to be used to connect adjacent bacterial cells and allow for genetic information to be exchanged between the two. Right now, a major concern in healthcare is that there are some genes that encode for pan-antibiotic resistance to every known antibiotic. And these genes have been located on plasmids, little chunks of DNA that can travel between bacteria on a sex pilus. Many gram-negative bacteria will have pili. A few gram-positive have pili. The pili in gram-negative bacteria is one of the reasons why gram-negative bacteria are, generally speaking, more pathogenic than gram-positive bacteria, because they are allowed to share their drug-resistant genes very quickly in a bacterial population amongst the bacterial cells through the sex pili. So, here we can see some fimbriae on the surface of the bacterium. They are very small, very hard to see, and are used for attachment. You would need to get really close, and I actually need to put on my cheater glasses to see these guys. You can see, if you adjust the contrast of your monitor, you can see some small, itty-bitty tiny lines coming away from the surface of the bacterial cells. Those are fimbriae, and they are attaching the cell to its substrate. And then we have these tubes connecting bacteria. These attach adjacent bacteria, and they are known as pili, and they are used to exchange cytoplasmic contents between adjacent cells. Also on the surface, or on the external part of our bacteria, we have the S layer and the glycocalyx. The S layer is made of a very special protein, the S protein, and it has many thousands of copies of a single protein linked over and over and again, repeatedly. You can think of the S layer as a suit of armor or a chainmail that will form around the bacteria. Our bacteria will only don their suit of armor or their chainmail when they're in a hostile environment. So if we have a stressed out bacterium, it will produce the S layer, but if the bacterium is in metabolically favorable conditions, it will not produce the S layer because it doesn't need to protect itself from its environment. The glycocalyx is something that I personally struggled with when I was an introductory microbiology student. Because it's made of a slime layer and a capsule, how do you differentiate the slime layer from the capsule? When do you draw a line and say this is clearly slime layer and this is clearly capsule? I don't know. They're poorly defined. One is denser than the other one. Generally speaking, all that goopy, gunky stuff on the outside of the bacteria, the slimy stuff, is called the glycocalyx. And it has lots of polysaccharides in it with a few glycoproteins. These polysaccharides act like a giant sponge. Here's some random trivia for you. Mucus, the stuff that you blow out of your nose when you have a cold, is actually a hydrated polysaccharide. Now, the slime layer, generally speaking, is loose and not very viscous. The slime layer part of the glycocalyx has a primary function of protecting against water loss and nutrient loss, whereas the capsule layer is much more viscous, much denser, and more tightly bound to the cell of the bacterium. The main function of our capsule is to allow the bacterium to form biofilms or colonies within an agar plate. So, here's a summary of the outside of the bacterium. We have the cell membrane. Outside of the cell membrane, we have that cell wall made of peptidoglycan. If the bacterium is in a hostile environment, it'll coat itself with the S layer of protection. And then there's that poorly defined glycocalyx that has the slime coating and the capsule. If we were to zoom in on this mucoid colony, this colony looks kind of like mucus on our Petri plate, and we were to form a negative stain on it, you could see that there's this big fat layer around the bacterium that's not absorbing the stain. That is the capsule that forms around the bacterium. So this white region around the bacterium is part of the glycocalyx of that bacterium. So our glycocalyx has multiple functions. I briefly mentioned biofilms. If we have a biofilm, we have multiple bacteria interacting with each other in different ways, sometimes multiple species as well, surrounded by a hard-to-penetrate slimy shell. Biofilms have very strong ramifications in healthcare. Any kind of persistent infection or persistent bacterial colonization within the human body typically is going to be a result of a biofilm. Classic examples include plaque on our teeth. Or if a patient receives an infection from a catheter, an intrauterine device, pacemaker, or any other foreign object that can be embedded in the human body, many times a biofilm will form on that foreign object. One of the main dangers of a biofilm is that it's very difficult for antibiotics to get to the bacteria in the center of the biofilm. The antibiotics can all, generally speaking, get to the bacteria on the surface, but because of that thick capsule, it's very difficult for the bacteria in the middle of the biofilm to actually be exposed to the antibiotics. So capsules protect bacterium from the external environment. So if that bacterium is producing a capsule, it's going to stick and form biofilms and, generally speaking, become pathogenic or become more pathogenic. Another plus of the biofilms for the bacteria is that it's hard to swallow or phagocytize a bacterium if it has a capsule. So our macrophages, our dendrites, and our cells of our immune system that, generally speaking, are going to go through phagocytosis struggle with phagocytizing bacteria that produce capsules. Some bacteria have such thick capsules they are actually designed to reproduce and grow within a white blood cell after being phagocytized. Here we can see a biofilm forming. We have our bare naked surface, and a few little bacteria start growing on it, and they produce their capsules. That glycocalyx becomes nice and thick, and the bacteria start to stick to each other, and then we have a really thick mass of slime and multiple bacterial species all growing together, and the bacterial species embedded at the bottom, deep down in there, are almost impervious to antibiotic treatments. Many times, if there is a biofilm formation, one of the recommended methods of treatment is mechanical removal, scrubbing the biofilm. And if this is inside of the patient's body, it is not going to be a pleasant experience. So, concept check. Which of the following appendages is not used for attachment? Is it the slime layer, the flagellum, the pilus or pilus, or the fimbriae? So, pause and rewind the video or flip back in your PowerPoint slides and get me the answer. 2, 3, 4, 5. All right, the answer is B, flagellum. Here we go. We use the flagellum for motility, but the fimbriae and pili are for attachment, and our glycocalyx is also used for attachment, particularly the capsule within the glycocalyx, and the slime layer as well. So flagellum is not used for attachment. Slime layer, pilus, and fimbriae are used for attachment. If you have any questions, post them on the discussion board or shoot me an email. Happy studies!